Genetic engineering has rapidly evolved from a niche research field into a transformative technology that redefines the boundaries of biology and medicine. In recent years, CRISPR-Cas9 has emerged as a powerful tool that allows for unprecedented precision in editing the human genome. This breakthrough technology is revolutionizing scientific approaches and prompting innovative discussions about its wider implications. Today, we delve into a multifaceted discussion that spans the technical mechanisms underlying this technology to the ethical considerations it invokes. As researchers continue to push the envelope, the prospect of generating superhuman traits, enhanced cognitive functions, physical strength, and longevity has become a subject of intense scientific debate. The conversation is not merely about scientific capability, but also about the responsibility that accompanies the power to alter fundamental aspects of human life. This script explores the evolution of genetic engineering with a focus on CRISPR, highlights the scientific advancements that have made superhuman capabilities conceivable, and critically examines the ethical dilemmas that these innovations pose. Through precise analysis and rigorous fact-checking, we aim to provide insights that resonate with experts and enthusiasts alike. By uniting scientific rigor with ethical foresight, we foster a balanced perspective that supports sustainable advancement for all. Genetic engineering, in its modern form, traces its origins back to the discovery of DNA's double helical structure and the subsequent development of recombinant DNA technology. Early experiments in bacterial transformation and gene cloning laid the foundation for more sophisticated methods that emerged over the following decades. By harnessing techniques from molecular biology, researchers were able to modify genetic material with increasing precision. The advent of polymerase chain reaction, PCR, further accelerated progress by amplifying target genetic sequences, thereby facilitating deeper investigations into gene functions. In recent years, the CRISPR-Cas9 systems captured the attention of the scientific community due to its simplicity, efficiency, and versatility. Derived from a natural defense mechanism found in bacteria, CRISPR works by utilizing RNA-guided nucleases to target and modify specific DNA sequences. Its design, which mimics an immune response, allows researchers to make targeted changes at predetermined loci in the genome with minimal off-target effects. Compared to earlier techniques, CRISPR offers a streamlined and cost-effective method that has democratized genetic research and enabled groundbreaking experiments across various fields, including medicine, agriculture, and synthetic biology. Notably, technical enhancements such as high-fidelity Cas9 variants and advanced guide RNA synthesis have further refined its accuracy and safety profile. Empirical studies have demonstrated remarkable success in correcting genetic mutations in cellular models, paving the way for potential therapeutic interventions. As a result, CRISPR has transitioned from an experimental tool to a promising candidate for clinical applications. This historical journey, marked by iterative improvements and rigorous validation, underscores the profound impact of genetic engineering on modern science and provides crucial context for current debates surrounding the creation of superhuman capabilities and the ethical challenges they introduce. This comprehensive background also sets the stage for advanced technical discussions on genome editing and its broader scientific implications, with detailed quantitative analyses. Within the framework of genetic engineering, the concept of superhuman capabilities has evolved from speculative science fiction into concrete scientific inquiry. Researchers are exploring the possibilities of enhancing human physiology by leveraging genetic modifications to extend lifespan, improve cognitive performance, augment strength, and enhance resistance to diseases. This endeavor is driven by quantitative research that measures gene expression profiles, metabolic pathways, and neural circuitry, all of which contribute to the possibilities of designing improved human traits. Experimentally, modifications to the growth hormone pathways and telomerase activity have shown promising results in preclinical models, indicating that controlled genetic alterations can result in measurable performance enhancements. Advanced technologies such as CRISPR facilitate targeted interventions by precisely altering genes responsible for specific traits. For example, changes in the brain-derived neurotrophic factor, BDNF gene, have been correlated with improved synaptic plasticity and memory functions, while modulation of the dystrophin gene could potentially address muscular degeneration. Researchers are also investigating epigenetic modifications that might alter gene expression without changing the underlying DNA sequence, thereby offering reversible enhancements under controlled conditions. Quantitative models and statistical analyses are essential in these studies to predict outcomes and assess risk profiles. Furthermore, advances in computational biology and bioinformatics are enabling detailed simulations of genetic modifications, enhancing our ability to forecast physiological changes. Sophisticated analyses, such as genome-wide association studies and quantitative trait locus (QTL) mapping, provide the foundation for linking specific genetic variations to phenotypic outcomes. This data-driven approach opens the door to personalized genetic enhancement strategies that could be tailored to the individual's genomic profile, optimizing both safety and effectiveness. As scientists decode the complex interplay of genes and environment, the promise of superhuman capabilities becomes increasingly tangible. These innovations, grounded in rigorous experimental evidence and statistical validation, inspire both optimism and caution regarding their application in human enhancement. This detailed inquiry into superhuman traits not only highlights scientific possibilities, but also serves as a precursor to the ethical and societal debates that must be addressed as these technologies mature. Summarizing the promise, empirical research and precise modeling quantify genetic variables to demonstrate the feasibility of human augmentation. Advances in molecular diagnostics and sequencing refine our understanding of genotype-phenotype dynamics, paving the way for personalized genetic interventions. Collaborative efforts across engineering, computational science, and clinical research continue to propel this transformative field forward. And these integrative approaches validate theoretical models and inspire progressive advancements in human genetic enhancement. Ethical debates surrounding genetic engineering, especially with the advent of CRISPR technologies, have intensified as the boundaries of human modification expand. One central ethical concern is whether society has the moral authority to reconfigure the human genome. Many critics argue that intentionally altering genetic material is tantamount to playing God, raising questions about the natural order and the potential hubris of assuming complete control over human evolution. These concerns are deeply rooted in historical experiences with eugenics and unauthorized genetic experiments, compelling the scientific community to proceed with caution. A second ethical dimension centers on the issues of consent and autonomy. When genetic modifications are applied at the embryonic stage, future individuals are subjected to irreversible changes without the ability to consent. This raises compelling questions about the rights of future generations and the extent to which current scientific capabilities should dictate inherited traits. Furthermore, the possibility of asymmetrical access to genetic enhancements may reinforce existing socioeconomic inequalities, creating a divide in society between those who can afford these technologies and those who cannot. Such disparities could exacerbate social stratification, ultimately destabilizing the fabric of societal cohesion. Additionally, ethical evaluations must consider the long-term consequences of genetic modifications. The introduction of superhuman traits may have unintended ripple effects on ecological balance, population dynamics, and human health. Experimental models, supported by robust statistical data, indicate that even minor alterations in gene expression can lead to unforeseen systemic effects over generations. Therefore, it is essential to incorporate rigorous risk assessments and ethical review processes before any clinical application of genetic modifications is pursued. Moreover, various ethical frameworks provide differing perspectives on these issues. Utilitarian approaches may justify genetic enhancements if they result in improved overall well-being and longevity, while deontological ethics emphasize the inviolability of natural genetic makeup and warn against instrumentalizing human life. The precautionary principle, as applied in many regulatory settings, advises restraint in the face of scientific uncertainty, reiterating the need for thorough ethical scrutiny and societal consensus. Finally, transparency in research and public engagement is critical for navigating these ethical dilemmas. Empirical evidence derived from quantitative studies should be openly shared with the community to foster informed debate and policy making. In aligning scientific ambition with ethical responsibility, the dialogue surrounding genetic enhancement must remain both inclusive and adaptive. By rigorously questioning assumptions and continually reassessing moral boundaries, researchers and policymakers can work together to ensure that advancements in genetic engineering serve the collective interest without compromising the intrinsic value of human life. Integrating these diverse ethical considerations requires a multidisciplinary approach that includes bioethicists, molecular biologists, policymakers, and social scientists. Comprehensive ethical reviews should be mandated at all stages of research to safeguard against irreversible consequences. Quantitative risk-benefit analyses and longitudinal studies are essential to understand the long-term impact of genetic modifications. By fostering open dialogue and establishing robust ethical guidelines, the scientific community can strive to balance innovation with responsibility, ensuring that progress benefits humanity while honoring fundamental moral principles. Genetic engineering's rapid progress influences not only scientific research, but also fundamental aspects of society. The ability to enhance human traits incites a reevaluation of what it means to be human, challenging traditional notions of identity and self-worth. As genetic modifications become more accessible, a redefinition of societal norms appears inevitable. Data from recent demographic studies illustrate potential shifts in workforce dynamics, healthcare distribution, and intergenerational responsibilities. By integrating advanced genetic technologies, society may experience both positive outcomes, such as improved public health, and negative repercussions, like the emergence of new social hierarchies. Ethical implications and socioeconomic factors are intricately linked. Quantitative analyses reveal that access to genetic enhancements might be concentrated among economically advantaged groups, potentially creating a bifurcated society. Historical parallels, such as the disparities noted during the Industrial Revolution, suggest that technological progress can widen existing social gaps if not carefully regulated. As these modifications become part of everyday healthcare and lifestyle choices, comprehensive policies must address issues of fairness and equity. Public opinion, shaped by scientific evidence and media portrayal, plays a critical role in influencing policy direction and social acceptance. Furthermore, cultural and religious perspectives add layers of complexity to societal discourse. Diverse belief systems interpret genetic interventions through varied ethical lenses, sometimes opposing the manipulation of what is considered sacred or predetermined. Philosophers and cultural theorists argue that altering genetic makeup may disrupt the established social contract, potentially leading to unintended psychosocial effects. Global surveys indicate mixed reactions, with some communities embracing technological innovation while others remain deeply skeptical. In the long term, the interplay between genetic advancements and societal structures could redefine global demographics. Societies that successfully integrate genetic enhancements may witness shifts in educational priorities, labor market demands, and healthcare systems. Such changes require robust, data-driven policymaking and proactive engagement with community stakeholders to ensure that benefits are evenly distributed. As innovative genetic technologies continue to emerge, they bring with them profound societal challenges and opportunities. Strategic planning, informed by rigorous statistical evidence and interdisciplinary research, is essential to navigating this transformation. Educational initiatives to improve genetic literacy and inclusive policy frameworks are vital. In harnessing these advancements, society must balance progress with ethical integrity to secure a future where technological benefits are accessible to all, and the essence of human diversity is respected and preserved. Let innovation and ethics always guide us steadily. Technical challenges in genetic engineering underscore the limitations of current methodologies despite significant advancements. Off-target effects, where CRISPR inadvertently modifies unintended sections of the genome, present a formidable obstacle by potentially inducing harmful mutations. Quantitative studies have documented off-target frequencies as high as 10% in some experimental setups, indicating that even small errors can compound over multiple gene editing events. Efforts to develop high-fidelity nucleases have reduced these risks, yet the precision remains imperfect. Further complicating the issue, mosaicism in edited organisms, where not all cells carry the intended modification, undermines the uniformity and predictability of therapeutic outcomes. In clinical research, this heterogeneity can translate into variable efficacy and unforeseen side effects, necessitating stringent quality control measures. The interplay of cellular repair mechanisms such as non-homologous end joining, NHEJ, and homology-directed repair, HDR, further contributes to editing inconsistencies that are not yet fully understood. Detailed statistical models are being developed to predict and mitigate these uncertainties, but inherent biological variability persists. Moreover, limitations extend beyond technical issues. Ethical, legal, and social constraints impose boundaries on acceptable research practices, which in turn limit the scope of experimentation. Regulatory agencies mandate rigorous preclinical trials to assess both efficacy and safety, often delaying the translation of promising therapies to clinical use. The potential for misuse in unregulated environments, including bioterrorism and unethical human experimentation, exacerbates concerns around the application of CRISPR technology. As such, careful oversight and continuous reassessment of risks are essential, underscoring the need for interdisciplinary collaboration. Ultimately, while the promise of genetic engineering remains high, its practical limitations necessitate a balanced approach, one that rigorously interrogates both its capabilities and its inherent constraints to ensure responsible progress. Comprehensive experimental validations and iterative improvements in gene editing protocols remain imperative for overcoming current limitations and unlocking the full therapeutic potential of genetic engineering swiftly. Global regulatory frameworks governing genetic engineering continue to evolve in response to rapid technological advancements. Today's policies are derived from decades of bioethical debates, historical precedents, and the lessons learned from previous scientific innovations. Regulatory agencies across different nations strive to balance the promotion of scientific progress with the necessity to mitigate risks. Comprehensive guidelines emphasize the importance of transparency, safety, and ethical considerations while accommodating the pace of innovation in technologies such as CRISPR. By harmonizing diverse regulatory standards, countries aim to facilitate international collaboration and responsible research practices. In practice, enforcing these policies poses significant challenges due to varying cultural perspectives, legal systems, and economic interests. Some countries have instituted highly precautionary measures that restrict experimental gene editing, whereas others adopt a more permissive approach to foster innovation. Quantitative assessments, such as risk-benefit analyses and cost-effectiveness studies, play a critical role in guiding policy decisions at national and international levels. Moreover, public consultation processes and stakeholder engagement are increasingly incorporated into the development of regulatory frameworks, ensuring that a broad range of viewpoints is considered before policies are finalized. Looking forward, the dynamic nature of genetic engineering calls for policies that are both flexible and adaptive. Continuous monitoring of scientific advancements, combined with iterative policy reviews, can help bridge the gap between innovation and societal protection. Collaborative efforts among governments, academic institutions, industry players, and international bodies are essential for creating a cohesive global strategy. Ultimately, robust regulatory oversight is imperative to ensure that the transformative potential of genetic engineering is realized in a manner that safeguards public health, promotes fairness, and upholds ethical standards. Regular international summits and expert panels are increasingly vital for harmonizing policies, sharing best practices, and updating regulations in line with emerging scientific data and ethical considerations. This ensures dynamic global governance indeed. Reflecting on the journey of genetic engineering, it is clear that CRISPR technology represents both a monumental scientific breakthrough and a source of significant ethical debate. The future of human enhancement depends on our ability to integrate scientific innovation with responsible oversight. As research advances, interdisciplinary collaborations will be crucial in addressing both technical challenges and societal concerns. By leveraging quantitative analyses and strict regulatory standards, the next phase of genetic engineering promises to unlock new realms of possibility while ensuring that ethical values are preserved. This balanced approach will enable society to reap the benefits of enhanced human capabilities without compromising our core moral principles. Ultimately, the path forward demands transparency, robust dialogue, and shared commitment to both progress and responsibility. In embracing the future, we combine cutting-edge science with ethical stewardship, ensuring that every breakthrough contributes positively to society's evolution. Let innovation and ethics always guide us.